All right, welcome back, Bio 153 students. And we're now going to be talking about the immune system. So, chapter 22 in your textbook, we've already covered the lymphatic system, and now we're going to be paying our attention to the immune system. So, the immune system is broken down into two sections. Today, we're going to focus on part one, which is looking at the innate defenses, and then eventually later on, we'll look at chapter 22, part 2 of the immune system, the adaptive defenses. So, of course, a big thing in our news today is, of course, the coronavirus. We're always talking about the coronavirus. We can't get away from COVID-19. And hence, we look at these innate defenses and how they do their best to try to go after viruses. And we'll look at some of these internal defenses today that are a part of this defense system. This is a picture here of the virus itself, of the coronavirus, COVID-19 right here, you can see. And so very important to do our best to stay safe during the pandemic. And of course, wear a mask is very important. Wash our hands and social distancing. All these things are important to reduce our chances of getting COVID. And of course, there's different tests out there that you can do. The swab test to see if you actually have the virus at that moment. Are you positive or are you negative? Or another way, of course, is the antibody test to see if you've had it at any given point. So important, especially in the medical field, to be safe, wear your mask, wear your face shield with the mask, wear your protective clothing, wash your hands. All those things are critical when we think of COVID. Okay, so when we look at the immune system, again, it's broken down in two major defenses. So our discussion today is looking at the innate defenses, and later on, part 2 of our notes, our attention will be towards the adaptive defenses. But our lecture today is focused on the innate defenses. So there are several different lines of defense. Our first line of defense, in case you're exposed to some type of foreign item, if you are exposed to a bacteria or a virus or toxins or whatever, that first barrier we have here, first line of defense, the surface barriers. So that would be the skin, mucous membranes, and we'll elaborate on this coming up here. And then we have our second line of defense, and that's going to be our internal defenses. And a lot of our focus today is looking at the internal defenses. And you can see many of them that are listed here. The phagocytes, the NK cells, which is referring to the natural killer cells, inflammation, antimicrobial proteins, and hence the fever. And also looking at the functionality of white blood cells. So a lot of different internal defenses come into gear when you are exposed to a virus like COVID-19, or again, exposure to bacteria, fungi, etc. They are also considered to be more of being non-targeted, non-specific types of defenses. And then again, part 2, we'll get into that, the adaptive defenses, and those are considered to be our third line of defense. And they're broken down into 2 major types of immunity. So we have humoral immunity, which focuses on the work of our B cells or B lymphocytes, and also looking at cellular immunity, which focuses on the functionality of the T cells. Again, T lymphocytes. And they're typically more targeted and take a little longer to react, in contrast to our innate system. So this is kind of a nice introductory slide here as we go forward. So first we're looking again at the first line of defense. So that's what the surface barriers are. So that first line here. So first line of defense. Again, when you are exposed to toxins or fungi or whatever it might be, viruses, etc. And you can see there's a variety of them here that we're going to look at today. So first line of defense, I know you love my handwriting. There's tears. So that's a nice safety barrier, which can help to wash away any irritants, microbes that might be exposed to the eye. Of course, we have our skin, very important, a nice physical barrier to entrance of the microbes. Sweat and oil gland secretions, also important. They can kill many bacteria. So that's always key. So a lot of different ones. Saliva, even that can help. Washing microbes from the teeth and mucous membranes of the mouth. So a lot of great examples here for us. Even the acidic pH can discourage growth of organisms. So that's key too. We have a low pH in our stomach, for an example. Other examples of the first line of defense surface barriers. We take a look at our respiratory tract, and here we have mucus, which is able to trap microorganisms. There's cilia, which is found in the respiratory tract, which are, again, cellular extensions, and they have the capability of sweeping away trapped organisms. So if you were to breathe in lots of particulates in the air and bring that in, again, the cilia can try to get these foreign items out of the body, and eventually a cough reflex can occur. The stomach, again, contains acid, hydrochloric acid, which can kill many of these organisms, which is important. Even in the large intestine, we have normal bacteria inhabitants there, normal bacteria flora, so that can keep the invaders in check. And even when we look at the bladder, the urine there, which can wash microbes from the urethra. So lots of great examples here that we find as surface barriers. But internal defenses we look at next. They kick into gear and are necessary if the microorganisms are very aggressive and may invade deeper into the tissues. And that's where, again, examples of these microbes could be bacteria, viruses, fungi, environmental toxins, etc. And that's where all these internal defenses come into gear. So the phagocytes, we'll talk about those a little later, along with understanding more about natural killer cells, the inflammatory response, antimicrobial proteins, we'll talk about those a little later. There's several different types there, which we'll get into, such as interferons and complement blood proteins. And then finally, we'll have a discussion on fever. What we're seeing here in this picture, again, this is a picture here of the coronavirus, COVID-19 right here. So we'll get a little bit of an idea of some of these internal defenses which can try to go after the virus. All right, the first internal defense, and it's important to know these internal defenses and their functions. We're first going to take a look at our first one here, which is phagocytes. Okay, phagocytes. So there are different types of phagocytes. The first example we're going to discuss today is looking at the macrophages. The macrophages. Macrophages, again, they're derived from monocytes. So if you remember way back when we had our blood chapter, we talked about the different white blood cell types, and one was monocytes. And I mentioned that they can evolve into the macrophages. So here they are. The macrophages are considered to be the chief phagocytic cell in your body. And there are two major types of macrophages in your body. We have free macrophages and fixed macrophages. So, free macrophages are able to wander through tissue spaces. And you look at the word macrophage, free, so free being the capability of moving around. And they're able to do that. They're able to search for cellular debris and foreign invaders throughout the body. So you can find them in your lungs, for example, when we think of alveolar macrophages. And then there are also what are known as the fixed macrophages. And they are considered to be the permanent residents of some organs. So fixed mean that they're typically more stationary. And that's the case with Kupffer cells in the liver and microglia found in the brain. This picture over here is kind of cool. I love this. It's showing the macrophage attacking a bacterium. So the white glob you see here, this is an example of a macrophage. And you can see it has these cytoplasmic extension arms that are going out to grab the bacterium because it's nearby. So here's the bacterium in the green. And here is part of that macrophage, these arms that extend out. They're eventually going to grab hold of the bacterium and bring it to the core of the body, and then ultimately break down the bacterium. So kind of a cool little picture there for you to take in. Other examples of phagocytes. So we have here, when we think of phagocytes, another great example, neutrophils. Very important white blood cell, the most abundant type of white blood cell in your body. And so they can act as a phagocyte too. They produce a very important chemical known as defensins. This antimicrobial chemical is key because it will eventually help with piercing the pathogen's membrane. So literally wounding the bacteria or wounding the virus. And so by doing so, it already has some damage, and then it can go in and do the final blow. Another example of a phagocyte are eosinophils. So they also can be phagocytic if need be. So here's a picture of the eosinophil here. Here's a picture of that neutrophil right here. Okay, so we're very soon going to take a look at the steps involved with phagocytosis. And looking at the term phagocytosis, it's referring to the engulfing of foreign solids by cells. The engulfing of foreign solids by cells. And these cells, of course, are referring to neutrophils, eosinophils, a macrophage. This picture here is showing a macrophage. This purple structure here almost looks like a monster or something under the bed, but it isn't, I promise you. So this is a macrophage here, And you can see it has these cytoplasmic extension arms that are able to go after the bacteria. It's going ahead and using these extensions to pull the bacterium towards the core of the body. And then eventually in time, the bacteria will be broken down. So that's what we're going to look at very soon here, the steps involved with phagocytosis. Okay, so now we're going to take a look step by step of this process of phagocytosis. So ready or not, here we go, right? So phagocytosis. All right, so based off this image, the green structures that you see here, so the green structures here that you see, they represent the pathogen. So the pathogen would be things such as bacteria that you might be exposed to, or viruses, okay? When we think of COVID, right? Coronavirus, COVID-19. Maybe you're exposed to fungi, perhaps. So things along those lines. And the structure in purple here is the macrophage, okay? So that's the phagocyte there. Macrophage, again, derived from white blood cells, derived from the monocyte, to be exact. So now we look at the steps involved of how we break down the pathogen, because that's what phagocytosis is all about, breaking down, destroying the pathogen. So, number one, here we have the phagocyte, the macrophage in purple. And it's going to go ahead and try to lure the pathogen, or perhaps cellular debris. And so when the particular pathogen gets very close, again, there are these cytoplasmic extension arms that can go after and grab hold of that particular pathogen. And when it gets very close to, I should say, there are these complement blood proteins, some actually that are on the surface, some that are actually on the surface here of the macrophage, and it's a sticky coating they have too. So when that pathogen gets really close and nearby, like if this was it right here, eventually it can get stuck on that surface too. So first it's a matter of luring that pathogen nearby. Next, the phagocyte, such as the macrophage here, forms what are known as the pseudopods. And so, again, the cytoplasmic extension arms come out, can bring hold of the pathogen. The sticky surface, because of the complement blood proteins here, can help with that, which is called opsonization. So we'll look at that real soon. So now we have our pathogens in green here, and they're going to go ahead and come into the core of the body of the macrophage. And what happens now is we're forming what's known as a phagosome, okay? A phagosome. What that is, you can see here, it's a phagocytic vesicle. So it's a vesicle that you see right here, where the pathogen in green is now enclosed within the body of the macrophage, enclosed in a vesicle. What's going to happen next is, now that we've formed the phagosome, lysosomes, which are very small organelles in the animal cell, will come together and will fuse with the vesicle and forms a phagolysosome. Okay, so we can kind of see that here. This is the lysosome right here. Okay, the structure here. It comes here, fuses with the phagosome, and we form now what's known as the phagolysosome. The lysosome itself contains these very acidic hydrolase enzymes. And so they're going to be very important to going ahead and digesting the actual pathogen. So that's kind of what we're seeing here. If we look at step number 4, the lysosomal enzymes So these acidic hydrolase enzymes will actually digest the particles. So they're going ahead, they're digesting the pathogen, and what you're left with is kind of a residual body remaining. And that's kind of what we're seeing right here now, is this residual body that's left. This is pretty much indigestible material. So what will happen next is this vesicle here has the remnants of the pathogen, if it was a virus or whatever. And this overall cell then, this vesicle, will make its way to the edge of the macrophage and will spill its contents out through a process known as exocytosis. So exocytosis of the vesicle as a whole, which will go ahead and remove the indigestible material and residual material. Okay? Because again, this material is indigestible and the material is then released through exocytosis. This is one process of an internal defense where we can go after and kill pathogens. Okay? So definitely important to go and review this process. To understand this process, there are several videos that you can look at. So this one here is looking at the step-by-step process of phagocytosis. And so you're more than welcome to take a look at that. And I guess we'll play it here for you to start off today for our first video of phagocytosis.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
All right, I hope you enjoyed that, and we'll go on to the next video. This next video is going to be showing you phagocytosis again, and ultimately eating of bacteria. So I hope you enjoy this. And our final video here is looking at phagocytosis, where a neutrophil is going after Staphylococcus aureus, which is a bacterium. This is kind of cute, so enjoy.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
All right. Well, I hope you enjoyed those videos, guys. The last one's always cute when you hear the belch of the neutrophil, because he ultimately did his exocytosis. So, all right. The next internal defense we're going to take a look at today is taking a look at the natural killer cells. Okay? The natural killer cells. The NKs, sometimes they're called. Okay? So, natural killer cells. So, these natural killer cells, their function is that they're able to go throughout the blood and lymph, so in the lymphatic system and cardiovascular system, going through those blood vessels, and they're ultimately looking for cancer cells and virus-infected cells. So they go after them. That's their key, to go after and destroy. So this picture here is very nice. So here we have two natural killer cells in the yellow working together as a team. And here we have a cancer cell in red. So imagine this being leukemia as an example. So natural killer cells, they do that. They'll go after cancer cells, typically speaking. Hence, they can be a big role player with patients that are taking their chemotherapy, but maybe they're also taking immunotherapy to fight off their particular cancer. And hence, an elevated count of NK cells can definitely help that in the body. So when they go after cancer, they have the ability, these NK cells, natural killer cells, to induce a process known as apoptosis. Apoptosis. Apoptosis is considered to be programmed cell death, also known as cell suicide. And this is a natural thing that happens in our cells normally when we think of mitosis and cell division. When we think of the stages of the cell cycle. And looking at the checkpoints that occur, okay, when the cells are past their functional lifespan. And that normally, our body just is programmed to do apoptosis. But not the case in cancer cells. They continue to live on, and they're way past their functional lifespan. Hence the danger of cancer. So the natural killer cells are able to do that. They can gain up on cancer cells and actually induce apoptosis on them. And they can also do that not only for cancer cells that I've been talking a lot about in here today, but they can do that with virus-infected cells too. So hence, that's their target, is going after these cancer cells and virus-infected cells. The case with the natural killer cells, the NK cells. Next, we're going to talk about another internal defense, which is the inflammatory response. And I'm sure you've all seen this at some point in your life. You've encountered the inflammatory response. So this is triggered whenever body tissues are injured. So this could be from intense heat, from getting a first-degree burn, on the beach maybe perhaps, or second-degree burn. You burn yourself on the oven or whatever. Or third degree, you fall into a fire. So a lot of us have experienced that. Or it could be even physical trauma. You get cut on the arm or the leg, right? Or like this person here, they stepped on a nail. So that's always a lot of fun. So I don't know if you've ever done that before, but I certainly have. And yeah, it hurts pretty bad, guys. Or perhaps it's an infection. You get infected. A bacterial infection or, infection, fungi, virus. So definitely not good. And why does the body then show inflammation? Why does that happen? You can see the reasons here. So, prevents the spread of all damaging agents to the tissues, disposes of cellular debris and those pathogens, and sets up stages for repair. So we probably, again, have all encountered the inflammatory response at some point in our lifetime. When the inflammatory response occurs, there are going to be 4 major signs of the acute inflammation. So 4 signs are also known as 4 cardinal signs. And these signs typically, you can see here, number 1, redness. So the area will be very red. So you think about that when you, get stung by a bee, right? That area is usually pretty red. Or first degree sunburn. Heat. So there's usually some heat in those areas. I mean, you definitely feel that if you've been sunburned before, right? Swelling. There could be swelling. Definitely with the bee sting, there can be swelling. Maybe even a little bit if you get sunburned. And pain. Oh my gosh, is there pain? When you get stung by those bees, those ground hornets or wasp, boy, that can hurt. Or you get that sunburn, you burn your back or whatever. Ooh, that shower the next morning. Ooh, yeah. Some people, if it's a really severe sprained ankle, could even have number 5 here, which is known as the impairment of function. Okay? So, how do these signs of acute inflammation come about? Well, they occur because of the release of inflammatory mediators. So, these very, very special, unique chemicals that are released in your body that lead to these signs of inflammation. Some of these would be things such as histamine, which can be released from mast cells. Kinins, they actually lead to inducing pain. Or prostaglandins can induce the pain. Or the release of complement. We'll talk more about that in a bit. Or even the release of cytokines, which can attract a lot of white blood cells to the area of the wound or the bee sting or whatever it might be. So the big thing to get off the slide is to make sure that you know your 4 major signs of acute inflammation. Redness, heat, swelling, and pain. All right, so we take a look at this chart here. I know there's a lot happening here, but here we have an example of tissue injury. We can see the picture here. So imagine the student that's working in the chemistry lab. They're taking general chemistry or organic chemistry, or maybe they are taking anatomy physiology and doing a lab, and you're working with glassware. And maybe you accidentally drop the beaker, and you pick it up and you cut yourself, and you're bleeding everywhere. So now you have that tissue injury. So how does the body respond to this? Well, first it would be the release again of these chemical mediators right here. Okay? So the overall release of these chemical mediators. And ultimately then they can play a big role because in time, many of them are going to lead to the development of these major acute signs of inflammation down here that we've talked about already, right? The heat, the redness, the overall pain and swelling. And again, there could be some possible temporary limitation of joint movement. This, eventually, the goal is this, healing. Some of these chemical mediators, however, like we mentioned, can attract a lot more white blood cells to the area of the wound, right? White blood cells, WBCs, right? So white blood cells, WBCs. So more white blood cells, that's key. You'll get a lot more white blood cells. So leukocytosis, elevated count of white blood cells. And eventually they're going to play a significant role too to the healing process. And we'll elaborate on that coming up here. But again, this is just an overview of how this process of inflammation takes place. Okay, so if you remember off to the side on that past slide we looked at, we talked about the idea of an elevated amount of white blood cells that might go to that area of the tissue injury. So we take a look at this process here known as phagocyte mobilization. Phagocyte mobilization. So a phagocyte such as a neutrophil or eosinophil or ultimately a macrophage will go ahead and migrate to the area of the injury. Mobilization is all about them moving, having that capability of going somewhere. This process takes about 1 hour after the inflammatory response. So we look at this step-by-step process. Number one, we have what's known as leukocytosis. Leukocytosis. This is where, again, we have an increase in white blood cells. And here we're just focusing on neutrophils. But let's say you have neutrophils that increase in number, and they enter the blood vessels nearby the wound, from bone marrow. The next process we look at, number two, is known as margination. Margination. And this is where neutrophils cling to the capillary wall. So we can kind of see this here. Here's neutrophil here. It's clinging to the capillary wall. And it's doing that because it's preparing to eventually leave the capillary and move to the area of the tissue injury. The next step in the process we have, number three, is known as diapedesis. Diapedesis. This is where the neutrophil will actually go ahead and flatten and will squeeze out of the capillary, literally through tiny little pores. Will squeeze out of the capillary. So that's what we're seeing right here. Okay, look at that neutrophil. It's squeezing out of the capillary and now moving through the tissue to the area of the injury. The last step in this process, number four, is known as chemotaxis. Chemotaxis, where the neutrophils will follow the chemical trail. So they literally will go ahead. They have, you almost think they have like olfactory receptors or something, and they actually can go ahead and find their way based off of chemicals released. They literally can find their way to the injury site. And then once they get there, maybe they did find some bacteria like in the green here, and literally they can do phagocytosis. So that's what we're seeing here with this cell here. The neutrophil is undergoing phagocytosis to eliminate that bacteria, because maybe someone did get injury from a nail, and the bacterium, the tetanus was there, and so there you have it. So very important to make sure to know these four steps involved with the process of phagocyte mobilization. So here are just more examples of the inflammatory response. So imagine getting bit by a spider. Definitely you show the inflammatory response. So look at this picture here of the large area where someone unfortunately has gotten bit from a spider potentially. And sometimes if you get bit by a spider, or perhaps you don't do a very good job of cleaning yourself from dirt and things, you potentially could be exposed to staph. This can happen very frequently in a hospital setting too, staph. and this can be very serious, can give you very serious illnesses from the infection, and it's very possible you could die if you don't take care of it. You might eventually develop a pus, which is a mixture of dead and dying neutrophils that you can see in the yellow, broken down tissue cells. And sometimes that core can even leak out, and that whole area is living in dead pathogens. So it's pretty serious staph infections. So very important to wash your hands, use anti-hand sanitizer, take a shower. All those things are important to get any type of bacteria you've been exposed to, especially in a hospital setting, to take care of yourself for sure. Because you never know. Another example of the inflammatory response, again, looking at bacteria. This is a pretty deadly one here. Maybe you've heard of the bacterium, the necrotizing fascialis. This is a flesh-eating type of bacteria. And unfortunately, you can see the picture here of this young lady here. She actually was exposed to this from a contaminated river in the south in Georgia, where she actually fell into this river on a high cope course with her girlfriends, hit a rock, and this bacterium unfortunately was in the river on this rock, and she was exposed to it. And it actually was very aggressive and literally starting to eat her flesh. So she had many different parts of her body amputated so she could stay alive. Typically, this type of bacterium usually needs to be in pretty warm water year-round. So you won't really find this in Michigan, but again, in the south, in inland lakes, where the water temp may not get below 55 degrees per se, you could easily find this in these inland lakes in Georgia, or even some river systems. So Georgia, Florida, Louisiana, Texas, Alabama, Mississippi, very possible that you could find it there, especially if your immune system is suppressed already. Because I remember hearing a story of an older guy who actually got it swimming with his family in inland lake in Florida and actually died from exposure. And in a matter of a couple days, he was already dead from the necrotizing fascialis bacterium that he unfortunately encountered in the water. So I guess I'm not going for a swim anytime soon in these inland lakes in Florida. I'll stick to the ocean. Okay, so the next internal defense we're going to talk about today are interferons. Interferons are a type of antimicrobial protein. Okay, interferons. There are three major functions that they do in your body. Number one, most importantly, they're antiviral. So they go after viruses and they help to isolate the damage that a virus might do on your body, such as COVID-19, for an example. They also can help with reducing inflammation if need be. They can also activate macrophages and mobilize natural killer cells if they need help, per se, going after the virus. There are three major types of interferons that we'll have in our body. There are the alpha interferons. Okay, so alpha. So right up here, this is alpha. That's alpha. So the alpha interferons, they're important for attracting, stimulating natural killer cells to help out, if need be, to go after the viruses and enhance resistance to viral infection. So that's what the alpha interferons do. So pretty important here. Number two, we have what are known as beta interferons. So a beta interferon. We can see that with the picture up here. This is what the beta looks like. So this is a beta interferon. Their function, the beta, is they slow inflammation in the damaged area. And then number three, we have the gamma interferon. So gamma interferon. Here's the gamma right here. So gamma interferon, they're going to go ahead and stimulate the macrophage activity. Stimulate the macrophage activity because they can definitely help out too, going after and destroying viruses. So important to know the functions of your interferons. Here we have a picture of how the interferons work going after a virus. So here we have a virus. We can see that right here. And this is a picture right here of the virus. So imagine this being COVID. So imagine that being the coronavirus, right? COVID-19 right here. So viruses, are they considered by definition to be alive? Do they have all the major functions to be alive on their own? What do you think? Yes or no? Are viruses considered to be alive? What do you guys think? The answer to that, are viruses considered to be a life form? The answer is no. They're not considered to be a life form. Okay? That's because they can't reproduce on their own. They have to use an organism's cells for reproduction. And that's what they do to our bodies. So like if you get exposed to the coronavirus, which is a very, very aggressive virus, they can come into your body, okay? Because you know, these viruses a lot of them are just airborne, like corona. They come in through your mouth. The coronavirus can come in through your nose, right? And they go ahead and go into your body. And we can see this here. They can go ahead and reproduce. They can use your DNA for reproduction. They can use your enzymes, okay? All of those things, your enzymes, they can use all those things to go ahead and reproduce. And then ultimately, we have two new viruses. So the viruses is reproducing in your body. COVID is reproducing in your body. So what can happen? Well, interferon, this antimicrobial protein we mentioned, well, it might have the capability here to sense that the virus is in the body, like COVID. And what can happen then is the cell here can produce more interferon molecules. And then ultimately the vesicle containing the interferon molecules can get to the surface of the cell right here. Unfortunately though, host cell 1 has been infected by that virus. Yes, it makes the interferon molecule, but will die by the virus. Many of these cells are dying, okay? The virus is trying to go ahead and destroy the organism. So here we have the interferons here, these molecules, and there might be a docking site right here. So you can see this here, the interferon can go ahead and bind to an adjacent cell, turn on genes that are antiviral proteins. And then eventually what can happen here then, right up here, the antiviral proteins, referring to interferons, can block viral reproduction. And that's the major task of these proteins, small proteins, the interferons, is they go after viruses and ultimately are going to hinder their ability to reproduce. So that way they can be isolated because then a natural killer cell can come in and kill them, or a phagocyte like a macrophage can come in and kill them. So that's what this picture is illustrating here, is the fact that the goal here is to hinder their reproduction, hinder their ability to reproduce, and blocking off sites where they can come into a cell to use the cell's overall machinery, such as DNA, to go ahead and use our DNA for reproduction. Okay, so moving on, we're now going to look at another very important antimicrobial protein, which is looking at complement here. Complement. All right, so complement is made up of approximately 20 blood proteins that circulate in the body in an inactive form. So going through your blood, okay, in an inactive form. And they're very important for going after and destroying microbes, microorganisms. Some of the major characteristics about complement, when we think of some of their important functions, they definitely can help out with the inflammatory response. They're very key for killing bacteria. So that's a major function for complement, is that they can kill bacteria. And they usually can do this using cell lysis, which we'll look at in a moment. And they also can be very important when we look at the non-Pacific and Pacific defenses in the innate and adaptive defense systems. So just some major characteristics here. Okay, we're now going to look at complement. Now, in order for complement to work in the body, complement, this protein here, complement blood proteins, need to become activated, okay, because they move around in the blood in an inactive state. So there's several different ways how they can become activated. The most traditional method is called the classical pathway. And so where you have an antigen-antibody complex which is formed. And antibodies, they can bind to invading organisms. The antigen would be a foreign item here, okay? So the antigen is anything that's considered foreign in the body. So maybe it's, maybe the antigen could be anything. It could be a virus. It could be a bacterium. So this complex is formed. And by doing so, then you can have some of these complement blood proteins that can come together to form a complex and become activated. And so that's the case here with the complement blood proteins C1, C4, and C2. They all then can become activated. When this happens, then it becomes almost like a waterfall effect, a cascade chain of events, where you eventually can have the complement C3 protein activated and maybe eventually the C3b complement protein activated. And then this can lead to another important function of complement, which is known as the process of opsonization. So opsonization is important for coating a pathogen surface, such as a bacterium or a virus or fungi, etc., which helps to enhance phagocytosis to kill the pathogen. So opsonization is very important. Now, another way that complement can become activated is one called the alternative pathway. And this can be more of a spontaneous activation. There are different stabilizing factors, no inhibitors on the pathogen surface. So that's another way how complement becomes activated here. And again, forming C3. You can eventually then activate C3a, and this can ultimately lead to enhancing inflammation. And that usually is important when we think of mosquito bites, bee stings. Again, many examples when we think of the inflammatory response. Other complement proteins activated. You can have C3b, and then you have a variety of complement blood proteins that form what's known as the MAC, the MAC. And the MAC right here is an abbreviation for the membrane attack complex. The membrane attack complex. And the goal here is you form all of these complement blood proteins, the MAC, ultimately it forms a pore. And actually forms pores on the surface of a bacterium. Okay, so pores on the surface of a bacterium. Okay, bacteria. Like E. coli, for example. If you have that in your body, you ate some contaminated food or something in the refrigerator. It ultimately, with these pores, they form these holes on the surface of the membrane of the bacteria, and then water can actually come into the bacterium, and the amount of water leads to pressure, and in time, the cell lysis is referring to ultimate destruction of the bacterium. So we'll be looking at that very shortly here. So there's a lot going on this slide, and the big things to get off of this slide here is to know the functions of complement, okay? That's the big things to get off of this slide. Know the functions, such as complement helps to enhance inflammation. Complement can play a role in phagocytosis because of opsonization, okay? And also a big function of complement is forming this MAC, these pores, which helps to ultimately kill bacteria. So those are some major functions of complement. I don't expect you to memorize all these complement blood proteins here. I mean, you can if you want, but this gives you more of an idea of how these proteins are turned on and what function they do. So we take a look now at a major function of complement, which is killing bacteria, going after a bacterium. So this picture here is showing how the complement comes together here, right? The membrane attack complex, the MAC, okay? So the MAC, there we go. Again, MAC, abbreviation for the membrane attack complex. So you can see how there's holes being formed here. The complement come together, forming holes on the surface of the bacterium on the membrane. Pores are being formed. This is setting up the stage for eventually ultimate death of the bacterium through cell lysis. So cell lysis, pretty much the cells being destroyed, splitting. Now we can see here, now because these pores were formed, the MAC, right, membrane attack complex, pores formed of complement. Now if the bacterium is exposed to lots of water, water easily can rush in to the bacterium. So we're kind of seeing that happening here, this massive influx of water. And the bacterium no longer can contain a constant internal environment as the water comes in. And there's lots of pressure now occurring with the water. And there's no bilge pump to get rid of the water, so definitely not a good situation here. And then ultimately, because of the amount of water pressure, the bacterium will burst. The idea of cell lysis, and ultimately the bacterium will die. So the bacterium will die. Bacteria will die, okay? So that's the big function for complement, is killing bacteria. To get a good perspective here again, here's a video which talks about
located on an
complement.
exposed surface, such as the capsule of a bacterium. Complement is activated and the
So
bacterium
definitely
is
you probably want to
eventually
take a look at
lysed.
that for sure.
Let's take a look at the
So I definitely
classical
encourage you to
pathway.
watch
It begins when
this
C1
video
complement
here.
proteins bind to antibodies on the surface
Good
of the
times for
invading
sure here,
cell.
so.
C1 then splits,
So definitely take a look at
exposing
that video.
binding sites for other complement proteins that interact in a complex cascade that releases chemicals that promote inflammation. Several of the complement proteins ultimately interact to form a channel or hole in the target cell's membrane. This channel is called the membrane attack complex. These pores ultimately cause the rupture of the target cell.
Okay, the last function we're going to take a look at in here today of internal defenses is fever. Okay, so take a look at the fever. So there's different levels of fever. If you have a slight fever, that's a sign you're fighting the virus, but it's that high fever which is very dangerous because it can lead to ultimate death, and it can lead to dangerous destruction, denaturing of enzymes, proteins in your brain being destroyed, so brain damage, so not good. Of course, a fever is definitely one big sign of having COVID-19 for sure. Some of the main characteristics of the fever: one is the idea of a systemic response to invading microbes. And white blood cells, leukocytes, and macrophages, ultimately exposed to the foreign material, they secrete what's known as pyrogens. So pyrogens, we look at that word, pyro means heat. So pyrogens, they're chemicals that reset the body's thermostat upward. So play a big role ultimately with the fever occurring. Again, a mild fever, you definitely want to be cautious, and a sign definitely you're fighting that particular virus you've been exposed to. But you want to keep an eye on that, because if it gets too high, very, very dangerous, severe, could lead to death. So we've all been here before, right? Johnny has the fever, and we're hoping we're going to be okay. We're holding on to our teddy bear in bed, trying to get better. But some of the benefits to the fever, as a sign the body is fighting that particular virus. Benefits of the mild to moderate fever. So you can see here. So, 1, an increased metabolic rate of tissue cells. So that can enhance the body's repair processes. And 2, the fever can inhibit microbe multiplication, so increase in microbes. And that's because items such as iron and zinc are found in low amounts, typically sequestered in the liver, and the microbes can't get to those then. So some benefits to the mild to moderate fever here. So very important as a whole. Okay, we're now going to review some of the important concepts that we've covered in the first part of our immune system chapter on the innate defenses. So, number 1, for review, which of the following are phagocytes that break down pathogens? Which of the following are phagocytes that break down pathogens? So just take a moment to look at your possibilities and what the right answer might be here. Okay, and the correct answer based off of these choices, hopefully you went with choice letter E, both A, B, and C. All of these are phagocytes that go after and destroy pathogens like viruses and bacteria and fungi. So that's the case with the macrophage, neutrophil, eosinophil. So hopefully you went with letter E there. Number 2, which of the following are acute signs of inflammation? Which of the following are acute signs of inflammation? So what would be the correct answer here? All right, and the correct answer here would be choice letter E, all the above. So choice letter E, all of the above. And, gotta love mosquito bites, right? Gotta love summer. Number 3 for review. Blank are antimicrobial proteins that are antiviral, reduce inflammation, and will stimulate macrophage activity. So what would be the correct answer here? What would be the correct answer here? All right. And the correct answer, hopefully you're thinking along the lines of letter C, interferons would be the right choice. Number 4, blank are large granular lymphocytes that police the blood and lymph for cancer and virus-infected cells that induce apoptosis. Blank are large granular lymphocytes that police the blood and lymph for cancer and virus-infected cells that induce apoptosis. So what would be the correct answer? All right. And the correct answer here, I would go with, how about choice letter D? Letter D. The natural killer cells. Natural killer cells. All right. Well, I hope you enjoyed lecture today.